Thank you for having me. I'm from the Regional Water and Environmental Sanitation Center situated here at KNUST. That's the Kwame Nkrumah University of Science and Technology, Kumasi, Ghana. I'm a PhD student studying for environmental sanitation and waste management option. So I'll be presenting this on an objective of my PhD titled Adsorption of sulfamethoxazole in aqueous solution using chemically activated carbon derived from Megathyrsus maximum, which I'll hereafter refer to as MM. So my outline will run through from introduction to conclusion. The motive for this study was based on a preliminary monitoring campaign done on waste stabilization pond effluent in Kumasi, Ghana. It was aimed at monitoring 3 known recalcitrant pharmaceutical contaminants in wastewater. That is the sulfamethoxazole, which I would shorten as SOL for ease of pronunciation afterwards. Then carbamazepine, diclofenac. And from our studies, we discovered that SOL was the most prevalent of the 3 analytes. So we concentrated and would want to find how would augment our waste stabilization pond. In Kumasi, we have waste stabilization pond. We wanted to evaluate. Here, it's a general knowledge that conventional treatment system cannot remove waste pharmaceutical contaminants. We, however, embarked on a 6-month monitoring campaign, which confirmed the general knowledge. So with the mind of exploring adsorption process as an additional treatment step, because this waste stabilization, one of them is used as an aquaculture and the other is used for farming activities. So with that mind of exploring an additional treatment system to remove sulfamethoxazole, we looked at local materials which will make the production of the activated carbon, lower the cost of the production of activated carbon. We not only did the monitoring campaign, we also surveyed the wastewater because there is a link between pharmaceutical contaminants and the presence of pharmaceutical contaminants and the presence of antibiotic resistant gene. And then it was interesting to find out that we isolated Klebsiella pneumonia, a multi-drug Klebsiella pneumonia, and Pseudomonas aeruginosa, which had abundant heavy metal resistant genes and antibiotic resistant gene. So the specific objective of the study were to produce and characterize activated carbon from MM using an organic acid, acetic acid, and a mineral acid, phosphoric acid. We also determined the operational parameters and optimize them. And then we characterize the batch process using the available Isotherm and kinetic model. We got our Megathyrsus maximum, which I mentioned I will call MM, from the vicinity around KNUST premises. They were washed, dried, and we impregnated with 40% phosphoric acid separately from 40% acetic acid at a ratio of 4 parts of acid to 1 part of the precursor for 24 hours. Thereafter, we carbonized at normal temperature. They were, after carbonization, we sieved with 1 millimeter to 2 millimeter sieve, and they were kept away. So we characterized using the point of zero charge, the Fourier-transform infrared spectroscopy, the scanning electron micrograph, and of course, we did perform the proximate and ultimate analysis. So for the time I have, I'll go through the slides. If we have questions, we'll ask. So these are the methods. For the optimization, we use the 1 factor at a time, although we also looked at the response surface methodology, but it's not captured in this talk. So for the proximate and ultimate analysis, it's reviewed activated carbon typical of lignos produced from lignocellulose material. I want us to focus on the point of zero charge. Hg here, please, I'll define, is the derived, activated carbon derived from phosphoric acid. And the Cg is the derived from acetic acid. So it had a lot of difference, disparity between the Hg and 2.4 points of zero charge. I would want to say here that the entire experiment removal or uptake of sol was largely controlled by pH. So hence I'm emphasizing on the point of zero charge. Every other parameters, like I earlier mentioned, were typical of what you get from lignocellulose material. The Hg had a slightly higher carbon content compared to the Cg. And looking at the same images, comparing CgA and HgB, B, phosphoric acid activated had more wider macroporous pores, which indicated that there was an intense reaction between the material, the precursor and the phosphoric acid. So these were the functional group, the aromatics and the oxygen-rich functional group, the hydroxide between 3,333 to 3,163. So again, they had similar functional group, meaning there was no much difference in what the acid could do except for the pore spaces. So this functional group, we observe, for instance, UCG means after adsorption with sol, and UHG means after adsorption with sol, which is the spectra in blue, and the spectra in green is before adsorption. So we can see a shift in the hydroxide group from before adsorption. It was in 3,186 wave number. It shifted to 3,163 wave number. So the shifts, and then we noted the disappearance in the C bond here at 1414, and it completely disappeared. Same thing for the CG. So we submitted or settled that this functional group were involved in the adsorption of sol. We looked at the parameters pH, time, contact time, the effect of adsorbent dose, the ionic strength, and the initial concentration. Basically, the process was majorly controlled by pH and initial concentration. As we can see in the graph for Hg, as the initial concentration increased in milligram per liter, the absorption capacity also increased. For the pH, as the pH increased, the removal efficiency decreased. And for CG, Hg, it achieved 100% removal efficiency at pH 3 and pH 5 and reduced to 72% at pH 8. We looked at 5 Isotherm model, fitting the experimental data with 5 Isotherm model, the Langmuir-Friedrich and Jovanovic monolayer 2-parameter model, and the Redlich-Peterson and Siebes model, which are the 3-parameter model. For the Hg, the data fitted well with the Redlich-Peterson with a correlation coefficient of 0.978, and then the CG fitted more the Jovanovic monolayer, which is a 2-parameter model. For the beta value here, which represents the non-linearity constant, is quite high. It's showing that this the uptake of sol was controlled by multiple interactions. Time will fail me to go in details. Like I said, pH played a major role. We speculated absorption interactions such as hydrophobic interactions. For CG, electrostatic interactions were involved, and the pi-pi stacking interactions were involved as well. Both Hg and CG contact time data fitted the pseudo first order kinetic model, showing that the reaction was mainly a physical adsorption that took place. This can be explained by the fact that, for instance, we concentrated on this HG in terms of looking at it as a potential for feed application, and we found out that we could regenerate it. So lending to the fact that it was physical adsorption, and the type of interactions that were involved, they were not strong bonds, they were weak bonds. Here, since we had in mind, the motive was to look at translation into the field, we compared with Norit GAC 1240 commercially activated carbon. And by just looking at these plates, here, these treatments in duplicates were subjected to HG. That means they were, by the way, these experiments, we used the 10 milligram per liter. So looking at these plates, A and plate B, for those applied to the absorbent, we can see that this is more clear than that of the Norit GAC. So for HG, we only looked at the regeneration for HG since that was of interest and it performed better. So for HG, it achieved a 97% removal efficiency, while the Norit GAC achieved 91% removal efficiency. Using deionized water, because all other experiment before now was we used deionized water, we found 100% removal efficiency, showing that they may be competing as expected in the real wastewater, or other contaminants may be competing with soil in the wastewater. Regeneration came out well. After first to second cycle adsorption, desorption cycles, HG maintained a 100% removal efficiency. So in concluding, both activated carbon displayed physical, chemical, and structural parameter typical of activated carbon made from lignocellulose material. The one factor at the time revealed that pH and initial concentration influenced the uptake of sol by the activated carbon. And HG matched Redlich-Peterson model, indicating that multiple interactions were involved, and compared to CG, which was modeled more with the Jovanovic monolayer model. Both absorbents were best described by the first order kinetic model, revealing a primary mechanism of absorption, revealing that the primary mechanism of absorption was physisorption. And then HG showed promising field application due to the rapid equilibrium achieved in just 15 minutes and its capacity for regeneration, and it competed favorably with Norit GAC. We had a lot of constraints in this study, majorly due to analytical instrument constraints. We are not able to use the environmental concentrations found in nanogram per liter and microgram per liter. We rather use milligram per liter, but we believe that these results can be translatable to, we could extrapolate it to those concentration. Furthermore, we couldn't carry out the adsorption study, which mimics the real life wastewater treatment system due to time limitations. So we'll be looking at these in our future studies. These are references. I want to thank RESC, which is funded by the Ghana government through the World Bank under the African Excellence Center, for providing a fund for this study. And my appreciation goes to my supervisors, Professor Helen, Professor Nana, Professor Isoke. And I'm thankful to Precious for conducting the scanning electron microscopy for this study. Thank you. Can have your questions now.
Yeah, thank you for the presentation. We do have time for maybe one or two questions.
I have a question from Huba. Can you talk a bit about the practical application of this work? Is it scalable? And is it reasonable to think that it would work well for other antibiotic compounds?
Okay, yes. So like the background, I started by saying that what motivated, we started a monitoring campaign. We didn't want to solve a problem that was not endemic here. There are a lot of research on this topic out there, but we looked at our end, and there is also true that the data for pharmaceutical contaminant occurrence, as well as even trying to remove them from our wastewater treatment system here in Africa is quite scarce. So that informed the study here. Yes, we looked at soil was most prevalent. The others were not prevalent. We only found them 1 or 2 months for a period of 6 months, but consistently. So I surveyed 4 sites. For the 4 sites surveyed, soil was consistently available. So our interest is in what is in our environment, what is endemic here, what is prevalent here. So I think we localized we try to solve a local problem with it. So for scalability, we had just done the batch test. I mentioned that one of the limitations was the fact that we didn't have time to conduct the colon adsorption studies, which probably form further studies, inform further studies. So with that, we'll look at probably the high retention time and trying to translate it to the field. So per the way HG performed, I think it's still scalable. We still, however, we still need to do more research, but this is what we could do at the moment. I don't know if I answered your question. So